Okay, let's unpack this. We are diving into one of the most fundamental systems in your body, this incredible network that takes all this raw data from the world and, translates it into awareness.
Exactly.
Our focus today is strictly on Chapter 18, General and Special Senses. We're aiming to give you a complete foundational grasp of the anatomy and physiology here.
And our mission is really to cover everything, from a simple pain receptor all the way to the incredibly complex fluid mechanics of your inner ear.
All through an auditory guide so you can actually visualize this stuff without needing the diagrams in front of you.
And that's the perfect place to start, because we have to define what this system even does. When that information, that raw signal, whether it's light or pressure or a chemical, first arrives at your central nervous system, we call that a sensation.
Just the raw data arriving.
Just the arrival. But the real magic, you could say, happens later. The perception is the conscious interpretation, the awareness of that signal. And that happens up in the cortex.
So sensation is the delivery, perception is the interpretation.
Okay.
And we classify these delivery systems into two main groups. First up are the general senses.
Right. Those are the ones distributed all over the body.
Think pain, touch, temperature, pressure, that kind of thing.
And then you've got the special senses. These are highly localized, super complex, and found only in specific organs. We're talking sight, hearing, balance, smell, and taste.
So what makes this entire network even functional is this concept called receptor specificity. This is the idea that a single receptor is really, really sensitive to one type of stimulus, and it pretty much ignores everything else. A touch receptor isn't going to fire because of a chemical.
Precisely. And the simplest form of receptor we find is the free nerve ending. They're basically just the dendrites of sensory neurons. They're common, and they monitor an area we call the receptive field.
And that receptive field size, that's the key takeaway for how well you can localize a touch, right?
It is, absolutely. I mean, think about the tips of your fingers or your tongue. The receptive fields there are tiny, less than a millimeter across, which gives you that incredibly precise localization.
That's how you can tell 2 points are touching you at once.
Exactly. Now, compare that to your back or your forearm. Those fields are huge, which makes localization much, much fuzzier.
We also need to get into how these receptors code the information. And this is where we get the two main functional types.
Yeah, we talk about tonic versus phasic. So tonic receptors are what we call slow adapting. They're basically always active, always generating a baseline signal.
Like the photoreceptors in your eye.
A perfect example. Or the receptors constantly monitoring your body position. They give you continuous feedback.
And then there's the other type, the fast adapting ones.
Those are the phasic receptors. They're normally inactive. They only fire when there's a change, signaling, the intensity and the rate of that change. They adapt really quickly.
So when we say adaptation, we just mean the reduction in sensitivity to something that's constant.
Right. And there are two types. If you say, put on a watch, you feel it immediately. But that signal drops off really fast. That's happening at the receptor. That's peripheral adaptation.
Okay, that makes sense.
But then there's central adaptation. Think about a strong, scented candle. You notice it right away, but within minutes, you don't smell it anymore, consciously.
Right, you've gotten used to it.
But the receptors in your nose are still firing. It's your CNS, your brain, that's actively inhibiting that signal. And it turns out that only about 1% of all incoming sensory info actually reaches your conscious awareness.
That's a massive filtering system, which brings us perfectly to the general senses. We classify them by what they sense: nociceptors, thermoreceptors, mechanoreceptors, and chemoreceptors. Let's start with the one for immediate survival, nociceptors, the pain receptors.
Nociceptors are generally free nerve endings, and their job is to detect tissue damage. They're most common near the surface skin, joint capsules around blood vessels, but pretty sparse in deep organs.
Which is why that deep organ pain can be so hard to pin down.
Exactly.
Here's where it gets really interesting. Nociceptors give us two very different types of pain. First, there's fast pain, or pricking pain, carried by myelinated fibers.
It's that immediate, sharp, and short-lived sensation.
Then you get the slow pain.
Right, carried by unmyelinated fibers. This is that burning or aching that starts a bit later and just lingers. Functionally, fast pain makes you pull your hand off a hot stove, while slow pain reminds you for hours that you did some damage.
And we absolutely have to talk about referred pain. This is the classic heart attack example, right? The pain is in a visceral organ, but you feel it somewhere on the surface.
Yeah, like in the upper chest or the left arm. It happens because the signals from both the deep organ and that patch of skin converge on the same pathway in the spinal cord.
So the brain gets confused.
The brain just gets confused. It's so used to getting signals from the skin on that pathway, it just assumes that's where the problem is.
Okay, next up, thermoreceptors.
These guys respond to rapid temperature changes. You find them in the dermis, muscles, the liver, and the hypothalamus. And they're phasic, so they adapt fast.
So you notice when you jump into a cold pool, but then you get used to it.
You do. And here's a little fact.
Yeah.
You actually have 3 to 4 times more cold receptors than warm receptors.
Interesting. Okay, the biggest and most diverse category here has to be the mechanoreceptors. These respond to physical distortion, stretching, twisting, that sort of thing.
And they break down into 3 big classes: tactile, baroreceptors, and proprioceptors.
Let's stick with the tactile group first.
Okay, they range from the simple, unencapsulated types like free nerve endings and the root hair plexus, which just tells you when a hair moves, all the way to these complex, encapsulated structures.
And those encapsulated ones are where the function gets really specialized.
Oh, absolutely. You have tactile corpuscles, or Meissners. For light touch, they're fast adapting.
Yeah.
Then for deep pressure over long periods, you rely on Ruffini corpuscles. And what's critical about Ruffini is that they are tonic. They barely adapt at all.
So they're for continuous monitoring.
Exactly, for things like maintaining your grip. And then you have the vibration sensors.
The lamellated corpuscles, or pacinian.
Right. These are big, layered structures that are amazing at detecting deep pressure and vibration, and they're super fast adapting. Perfect for detecting a pulse or some other transient event.
Moving deeper into the body, we have the internal stretch sensors, baroreceptors.
Think of these as your body's internal pressure gauges. They're vital in the carotid and aortic sinuses for monitoring blood pressure, but you also find them monitoring your lungs, your bladder, your digestive tract.
And finally, the sense that lets us stand and move without having to look at our own limbs, proprioceptors.
These monitor joint position, tension in your tendons with Golgi tendon organs, and muscle length with muscle spindles. And since you can't just occasionally be aware of where your limbs are, these receptors are unique because they generally do not adapt.
It's just a non-stop, constant stream of feedback.
Continuous, moment by moment.
All right, and briefly, the chemoreceptors.
These are specialized neurons monitoring tiny changes in dissolved substances. The key spots are the carotid and aortic bodies, where they're tracking pH, CO2, and oxygen to adjust your breathing and heart rate on the fly.
Okay, let's shift gears now to the special senses. Starting with olfaction, or smell, the organs are way up high in your nasal cavity. You've got the olfactory epithelium and the lamina propria, which has the glands, Bowman's glands that secrete all that mucus.
And the receptor cells themselves are actually modified neurons with up to 20 cilia that project into that mucus. The system is, it's just ridiculously sensitive. The sources say as few as 4 molecules can set one off.
Wow. And the signal then passes through the cribriform plate up to the olfactory bulbs.
It does.
And this is the key fact, the one that gives smell its huge connection to memory and emotion. So listen up. Olfactory sensations are the only sense to reach the cerebral cortex without first synapsing in the thalamus.
That direct, unique bypass is why a certain smell can trigger such a powerful, immediate emotional response. It has a direct line to the limbic system.
And on a practical note, this system declines with age.
It does. The number of receptors goes down, which contributes to a reduced sense of flavor in later life.
Which brings us right to gustation, or taste. The receptors are in taste buds, which are nestled in the papillae on your tongue.
And inside those buds, you have gustatory cells with these little taste hairs that stick out through a taste pore. We now recognize 6 primary tastes: sweet, salty, sour, bitter, umami, that savory flavor from amino acids, and even water receptors in the pharynx.
And there's a huge difference in how sensitive we are to these, which goes right back to survival.
Absolutely. We are thousands of times more sensitive to bitter and sour compounds than to sweet ones. It's a massive evolutionary advantage. So many poisons and toxins are bitter, and strong acids can damage tissue. It's an immediate warning system.
And the final piece here, why does food taste so bland when you have a cold?
Because what you think of as flavor isn't just taste. It's overwhelmingly smell. When your nasal passages are blocked, you lose that huge amount of information from olfaction, and all you're left with is the basic sweet, salty, and texture from your tongue.
Okay, let's navigate the anatomy of equilibrium and hearing. Starting with the outside, the external ear, you have the auricle, which funnels sound into the external acoustic meatus, ending at the eardrum or tympanic membrane.
And you have cerumen, or earwax, in there for protection.
Then you get to the middle ear, which is this air-filled cavity. It connects to your throat via the auditory tube or eustachian tube.
Which is so important for equalizing pressure on either side of the eardrum.
Right. And inside you have the three tiny bones, the auditory ossicles, the malleus, anscus, and stapes. They transmit the vibration to the inner ear.
And they amplify it. The eardrum is about 22 times larger than the oval window where the stapes sits, so that force gets concentrated and amplified significantly.
And there's even a built-in protective mechanism.
Yeah, two tiny muscles, the tensor tympani and stapedius, contract in less than a tenth of a second to dampen really loud sounds and protect those delicate inner ear structures.
Okay, now for the most complex part, the inner ear. This houses the receptors for both balance and hearing.
And all those receptors are inside the fluid-filled membranous labyrinth, which is floating inside the bony labyrinth. The bony part is divided into the vestibule and semicircular canals for balance and the cochlea for hearing. And all the receptors are a type of hair cell.
So let's break down equilibrium or balance first.
Right.
How do we sense turning our head?
That's the job of the semicircular ducts. When your head turns, the fluid inside the endolymph lags a bit, and it pushes against this gelatinous structure called the cupula.
Like a swinging door.
Exactly like swinging door. When it bends, it stimulates the hair cells, telling your brain about rotation.
And what about gravity or accelerating in a straight line, like in a car?
That happens in the utricle and saccule. Their hair cells are embedded in a gel that's topped with these heavy little calcium carbonate crystals called statoconia. The whole complex is an otolith.
Like a little weight sensor.
It's a biological inertia sensor. When you tilt your head or accelerate, that heavy mass shifts, it bends the hair cells, and it signals the direction of the movement.
What's fascinating here is the mechanics of hearing inside the snail-shaped cochlea.
So the cochlea has the cochlear duct, which is sandwiched between two other chambers. When the stape pushes on the oval window, it creates pressure waves in the fluid, and that pressure has to go somewhere, so it's relieved by the flexible round window at the other end.
And it's those pressure waves that do the work.
They do. The waves distort the basilar membrane, which is the floor of the cochlear duct. This pushes the hair cells of the organ of Corti upwards, so their little stereocilia brush against the stationary tectorial membrane. That's the stimulus.
And pitch is all about location.
Precisely. High-frequency sounds distort the membrane right near the beginning by the oval window. Low-frequency sounds travel much farther down toward the apex of the cochlea.
All right, finally, let's move on to vision, the sense we rely on more than any other. Protection is key, starting with the lacrimal apparatus.
The lacrimal gland makes your tears, which contain an antibacterial enzyme called lysozyme. The tears wash across the eye and then drain through little openings called lacrimal puncta and eventually end up in your nasal cavity.
That's why your nose runs when you cry.
That's exactly why.
The eye wall itself has 3 layers. The outer fibrous tunic, the white sclera and the clear cornea, the middle vascular tunic, and an inner neural tunic, which is the retina.
And that vascular tunic is highly specialized. It includes the iris, the colored part, which controls your pupil size, and it also holds the ciliary body and the choroid.
The internal fluid balance is also critical. The big back cavity has the jelly-like vitreous body, but the anterior cavity has a circulating fluid, the aqueous humor.
Aqueous humor is a bit like CSF. It provides nutrients, and it's constantly draining through the canal of Schlemm. If that drainage gets blocked, pressure builds up, and that leads to glaucoma.
Which can cause blindness.
It can. The pressure damages the retina and the optic nerve.
The lens is the final mechanical piece for focusing.
Right. Its shape is controlled by the ciliary muscle.
Yeah.
To focus on something close, the muscle contracts, which actually relaxes the tension on the suspensory ligaments. The lens is elastic, so it naturally gets thicker and rounder.
And when that lens loses its transparency with age, that's a cataract.
That's a cataract, correct.
So the final destination for light is the neural tunic, the retina. This is where light becomes a neural signal.
And it contains 2 types of photoreceptors. We have about 125 million rods, mostly in the periphery. They're for black and white vision, incredibly sensitive to dim light.
And then the cones.
About 6 million cones, concentrated in the macula lutea. They give you sharp detail and color vision. And the absolute sharpest point of your vision is the fovea centralis, which is packed almost exclusively with cones.
So the signal goes from the rods and cones to bipolar cells and then to the ganglion cells.
And the ganglion cells are the only ones that generate true action potentials. They're axons all bundled together at the optic disc, which is your blind spot, to form the optic nerve.
And the visual pathway has that really critical crossover.
It does. The optic nerves meet at the optic chiasm, where about half the fibers cross to the other side of the brain. This ensures that each hemisphere gets a complete, composite visual field.
From there, it goes through the thalamus to the visual cortex in the occipital lobe.
And don't forget that little branch of visual info that goes to the suprachiasmatic nucleus in the hypothalamus. That light information is what synchronizes your body's circadian rhythm.
So what does this all mean? We've successfully navigated the immense complexity of your sensory world, from a simple free nerve ending all the way to the high-res processing unit that is the retina. We've covered why you ignore your watch, why pain can be misplaced.
And the evolutionary advantages of our most basic tastes.
Yeah. We've really provided that high-speed, comprehensive breakdown of the entire sensory systems chapter.
And to tie it all together, just consider the constant integrated activity happening inside you right now. Your proprioceptors are reporting joint position. Your visual pathways are sending light cues to set your internal clock. Your auditory system is registering microvibrations even when you feel like you're in total silence and stillness.
It never stops.
It never stops. So
Yeah.
how fundamentally does this constant subconscious sensory stream dictate not just your awareness of the world, but your entire internal metabolic and psychological state? That's something to really consider the next time you try to ignore a sensation.